Hi everyone. In last class, we discussed about one of the computational method to predict protein structure is known as homology modeling. In today's class, we will be discussing about other two methods to predict protein structure known as fold recognition method and ab initio method. First of all, we are going to discuss about fold recognition method. Fold recognition method is also known as threading method. It mainly depends on the protein folding. We can define protein folding as arrangement of the secondary element of the molecule which are related to each other. There were about 5000 protein folds in nature. So, in fold recognition method, we are going to get the best fit of the sequence to our target sequence. And generally, it is used when the homology modeling fails to predict the protein structure. We can say if the sequence similarity or identity is less than 30%, in that case, protein folding or fold recognition method is preferred. Now, we are going to look at the how fold recognition method going to occur. So, we have to mount the target sequence or protein sequence to our template sequence by using SCOP database. It is known as fold recognition database or fold structure database. And then for each step, we have to place our sequence along the template sequence at each position. Then we have to find the folding ratio for each fold that we are going to study. Generally, fold recognition method follows four main criteria. The first one is hydrophobic collapse. The second one is local interaction which stabilize secondary structure, and then secondary structure interact with the motif formation. Finally, the motif formation aggregate to form the final protein structure. There are two main algorithms for fold recognition method. One is profile based method, another one is pairwise energy based method. In pairwise energy based method, we will be searching for the structural fold databases in order to get the best structure by looking at the energy values. Here, dynamic programming and heuristic approaches will be considered, and then energy will be calculated for the raw model, and the lowest energy fold will be considered as the best fold for the structural prediction. Whereas in profile based method, profiles will be generated initially by comparing the relative protein structure, and then we will superimpose our structure to get the corresponding residues. We will also consider the secondary structure type, polarity and hydrophobicity, and also the structure which is predicted in this method. If it is not present in the protein folding library, then the method fails to predict the proper structure. Now we are going to discuss the steps involved in threading method. Initially, we will have unknown protein sequence or amino acid sequence. Then we will perform BLAST program in order to find the identity and similarity of a target sequence to the template sequences. Once we get the template sequence, we will get the template structure from the PDB databases. After that, the template structure will be taken to the fold database in order to find the fold templates. Then the fold template which we got from the fold library will be compared to our target structure, our target sequence, and each location of the target will be compared to the template sequences in order to find the best fold sequences which help us to predict the protein structure. Now we are going to look at the pros and cons of threading or fold recognition method. It will give you a good result, but then the experimental properties will be more accurate compared to the amino acid sequence similarity matrices, and also it will be very effective and implementative process. And then this method is used only when we have a proper protein structure which is already present or determined initially in the databases. If not, this method will not be preferred. And also it helps us to give you the sequence similarities which is not given by the sequence methods. Now we are going to look at the third and the final computational method to predict the protein structure, that is ab initio method. So ab initio method is preferred when there is no sequence similarity or very less sequence similarity of our target protein with our template protein. This ab initio method is very difficult process where proteins are folded into a different conformation. This method is mainly depends on the thermodynamic principle proposed by Atkinson. He stated that the native structure of the molecule is mainly depends on the global free energy minimum under a given set of conditions. And also the ab initio method approaches are Rosenta, I-Taser and touchstone. The mostly preferred one is Rosenta and also I-Taser. And this method is mainly depends on the Monte Carlo algorithm. This method is also known as free energy modeling or physics-based modeling, or also it's known as de novo modeling. This method is mainly depends on three main factors. One is accurate energy function, second one is conformational search, and the third one is native-like model. And also when compared to other two methods, this method does not need any protein sequence databases to compare our target sequence to the template sequence. There are two main steps in ab initio method. One is optimization, another one is simulation. In optimization, we will define a basic model and then we'll define a function mapping structure and then we'll solve computationally in order to find the global minimum value. Whereas in simulation, we will initially define or find the initial model by looking at the energy and force values and then accurately simulate the dynamics of the system, and then finally we will arrive at the native structure of the protein. Now we are going to look at the steps involved in Rosetta program in order to find the protein structure. There are three main steps. They are, we have to find the fragments first, and then we have to assemble fragments into models, and then we have to find the best model out of it. So in the first step, we will be considering about 25 to 200 fragments of monomer, trimer and nanomers, and then the fragments are ranked based on the sequence similarities and also similarity between the predicted and also known protein structures, and then the fragments of low quality will be discarded in this step. And in the second step, we will be doing local refinement strategies where three main analysis will be done. The first one is we will be randomly doing torsion angle perturbation, and the second one is selection of globally non-perturbed fragments, and then finally we will be doing adjacent pi angle and psi angle variation will be calculated. In the last step, we will be selecting the best model by generating the population decay structure of 10 to the power of 3 to 10 to the power of 5, and we will be filtering in order to avoid biases, and then we will be doing cluster analysis. Finally, full atom refinement will fetch you the best model with low energy value. Now we are going to look at the advantages and disadvantages of ab initio method. The advantages of this method are, it is used to find the protein folding, it also helps us to find the protein misfolding, and also this method does not need any homologous protein for its comparison, and it also used to find the de novo analysis for protein designing, and is also used to find the models of new folds. These are the advantages of the ab initio method. Now we are going to look at the disadvantages of this method. The disadvantages of this method are, this method is not preferred for the proteins of greater than 100 amino acid residues, only preferred for smaller proteins, and it is very experimentally expensive, and it is very time consuming method, and it's also the accuracy of this method is very low. Till now we saw three computational methods to predict the protein structure. They are homology modeling, fold recognition method and ab initio method. So homology modeling will be done for the proteins with the sequence similarity of greater than 30% with the template structure and with the target structure. If your protein similarity is less than 30%, then homology modeling will not be preferred. And in homology modeling, we use PDB databases for template structure recognition with the comparison of target sequences. Whereas in fold recognition method, it mainly depends on the protein folding. So in fold recognition method, usually the similarity between template and protein structure, if it is less than 30%, then we can do a protein structure prediction in fold recognition method. So in fold recognition method, the proteins which are failed to perform in homology modeling can be determined in fold recognition method. That is, the similarity between template and target sequences which are less than 30% can be compared in fold recognition method. And the final method is ab initio method. This method is entirely different from other two methods, that is homology modeling and fold recognition method. This method is mainly depends on the physics law, and also in this method the protein structure will be determined from the scratch, and also it's mainly based on the energy value of the final protein structure. We came to the end of this topic, and also we came to the end of this video lecture series. In this lecture series, we learned about 8 topics. They are structure of protein, Ramachandran plot, methods of protein structure prediction, X-ray crystallography, NMR spectroscopy, crayon electron microscopy, homology modeling, fold recognition method and Initio method. Hope you understand about the basic concept of protein structure prediction. In future, we will be seeing about more interesting topic in biotechnology field. Thank you all.